In today's video, we are going to look at the physiology of muscle contraction. So basically, the mechanism that outlines the various processes for a muscle to simply contract. And when we talk about muscle contraction, we are basically looking at the muscle's ability, first of all, to be stimulated. And when it's stimulated, it undergoes certain mechanisms to lead to muscle contraction. Again, when we talk about muscle contraction, we are focusing on the muscle fibers' ability to shorten in length. We'll look at that pretty shortly. So these are the three stages muscle contraction would have to go through. So first of all, there should be electrical excitation of a muscle fiber. Remember, I said that the muscle has to first of all be stimulated. So we'll look at that under electrical excitation. Then once the muscle is stimulated or excited for that matter, that excitation will couple the process of contraction. We call it excitation-contraction coupling. Then the final stage is for the muscle fiber, as I said, to contract. And this is as a result of the sliding filament mechanism of the myofilaments. The myofilaments are the actin and the myosin filaments. So they will cause the entire length of the muscle fiber to shorten. When it shortens, then the muscle has contracted. When it begins to widen, it means that the muscle is relaxing. But here we are talking about muscle contraction. All right, let's dig right in. So look at this structure. This particular structure here is an axon terminal, as has been labeled here, the axon terminal of a somatic motor neuron. And this axon terminal is terminating on a muscle fiber. So an axon terminal of an axon or of a neuron that is synapsing with the motor end plate. And when we say a motor end plate, as you can see here, we are talking about the side of the muscle, which if we are talking about the neuron and the neuron synapsing together, this will be a postsynaptic membrane. But for a postsynaptic membrane of a muscle fiber, we call it a motor end plate. So take note of that. A motor end plate would have a motor neuron synapsing with it at that stage. So you can also notice that this axon terminal is having some vesicles inside them. Then there are some neurotransmitters that have been released here, and there's an activity going on. So we want to describe exactly what is going on here. First of all, you can see this point 1. So this point 1 would stand for the arrival of an action potential, and I will cover the process of action potential in another video. So action potential arrives at this axon terminal. Now the purpose of this action potential is to cause a particular gate, which is not shown in this picture. So that gate is called voltage-gated calcium ion channel. Voltage-gated calcium ion channel. So when that voltage-gated calcium ion channel, which is sensitive to the arrival of the action potential, it would cause the influx of calcium ions. So when calcium ions enters into this particular axon terminal, then it will stimulate these vesicles. So these vesicles are containing the neurotransmitters. It could be any neurotransmitter at all, depending on the kind of message that is being communicated. So let's say it is acetylcholine, as has been indicated here. This ACH is acetylcholine. So the arrival of the action potential causing voltage-gated calcium ion channels to open, and then the subsequent influx of calcium ions into this axon terminal is going to cause these vesicles to begin to migrate to the end of this neuron or axon terminal. Now, when they migrate to the end or the membrane of this axon terminal, they would undergo what we call vesicular docking. So that is exactly what these structures have done. So these are vesicles that have docked onto the membrane of this axon terminal. Then by way of exocytosis, they release their contents into this synapse. Okay, so this entire structure is a neuromuscular junction. A neuromuscular junction is a neuron and a muscle fiber forming a junction, and there's a synapse in between them. The synapse is the gap. All right, so over here you can see these neurotransmitters released onto this synapse. Another point that would continue here is that these neurotransmitters are going to locate their specific receptors on the motor end plate and bind to it. In this case, they will bind to their specific receptors, and these receptors are going to be ligand-gated receptors or ligand-gated channels. So these channels are sensitive to the binding of a neurotransmitter. An example is what is happening here. So here, when the neurotransmitters bind to this channel, it would couple the influx of sodium ions. So sodium ions that are already inside this extracellular fluid medium will now begin to enter into the motor end plate. When they are doing that, remember that they are going to continue the process of action potential. So the action potential that arrived at this axon terminal has now been transferred to this motor end plate, and I've described how that happened. So once action potential or this particular this particular muscle is stimulated, that is to say that it has received the action potential. This action potential is going to travel down this structure called T-tubule. The T-tubule is found within the muscle fiber. Now take note that this wave of action potential is going to flow down the T-tubule. And when we say action potential is flowing down, basically we are saying that ions are entering into the cell and exiting the cell. So they go through 3 stages: depolarization, repolarization, and then hyperpolarization. That cycle continues on and on like that. And I've said that I'll cover that in my next video. So the arrival of the action potential goes down the T-tubule and it reaches the base of the T-tubule in this manner. Now the purpose of this is that there is another sensitive receptor here, which is called DHP. Now this DHP will only open, the full name of DHP is dihydropyridine receptor. It will only open only at the arrival of this action potential down the T-tubule. So when the action potential gets to this T-tubule, it will then cause the opening of this dihydropyridine receptor. And as you can see, this DHP is mechanically coupled to another receptor called ryanodine receptor, which is the RYR here. So RYR stands for ryanodine receptor. And this special receptor encloses this sarcoplasmic reticulum. And as you would imagine, this sarcoplasmic reticulum stores calcium ion within the muscle cell. So up until now, these calcium ions are enclosed within the sarcoplasmic reticulum. But then because of the arrival of the wave of action potential down the T-tubule, it will cause the opening of this ryanodine receptor. The opening of ryanodine receptor will also mechanically couple the opening of the ryanodine receptor, which would open this gate. Then calcium ions will begin to flow into the cell. So now look at what is happening here. The same picture is what is shown here. So we have calcium ions that are inside the sarcoplasmic reticulum, which are now going to flow into the membrane of the muscle cell. So take note, calcium ions are going to increase and it will flow down into the muscle fiber. So going back to this picture again, we have calcium ions that are entering into the muscle fiber. Now over here, notice that this is actin and this is the myosin fiber. The actin is a thin filament and then the myosin fiber is a thick filament. In another video, I'll cover only on this actin and the myosin thick filament. So I want you to visit that video to really understand the anatomical structure of these 2 filaments. That will give you much more perspective into what is happening here. But basically what would happen next is that the purpose of the influx of calcium ions from the sarcoplasmic reticulum is to bind to this structure or this protein called troponin. So this troponin has 3 subunits. We have troponin I, troponin C, and the troponin T. As I said, I'll cover a video on that. Specifically, the calcium ions will bind to the troponin C. Then when the calcium ion binds to it, because it is a protein, it will undergo conformational change, which will cause the unwinding of this tropomyosin. The purpose of the unwinding of this tropomyosin is because up until now, this tropomyosin has covered all the active binding sites on this protein structure called actin, meaning that this myosin head cannot bind to this actin to bring about muscle contraction. But because of the arrival of calcium ions binding to the troponin C, causing the unwinding of this tropomyosin, there is the exposure of the active binding sites of the actin. And now, these myosin heads can now bind to it. When they bind to it, they will now perform this power stroke that is described here. And I'll cover this also in another video. So technically, these myosin heads can now bind to the active binding sites. As you can see here, the myosin heads bind to actin, forming a cross bridge. Then there will be a power stroke as a result of the release of a phosphate group from ATP. If all of this doesn't make sense, watch the other video I'll be releasing on the power stroke. But then this will cause the pulling against of the myosin head to the actin, causing the shortening of the length of the entire muscle fiber. So this is what we talk or we describe as muscle contraction. Doing a recap of what I have said, this point 1 illustrates the arrival of the action potential. Then this arrival will cause the opening of this DHP receptor, which is called dihydropyridine receptor. That will couple this ryanodine receptor channel to open, causing calcium ions to flow into the muscle fiber. Once calcium ions are present, the calcium ion binds to troponin C, causing the unwinding of what? The tropomyosin, exposing the active binding site on this actin. Then the myosin head can now bind to bring about the power stroke. So this brings me to the end of this video.